Okay. So I'll leave you with the presentation, but I'll just make a few comments about it. Many of you will have been here for the first session on Data Warrior, but I can see that we have a few new people as well. If you have any questions, put them into the questions box and I'll relay them to Garrett. Garrett is going to give a rather more informal presentation on PyMOL and very much requests for audience feedback and interaction. So very much thanks to him. This would have been a physical meeting normally, but the advantage of having it as a virtual meeting is that we can actually have audience from all around the world and probably orders of magnitude higher number of people. So with that, I'll hand it over to you, Garrett, and I'll switch off my video so that I'm not there as a distraction.
Okay, cool. Thanks. Thank you, Chris. So as Chris said, I'm going to make this as interactive as we possibly can. And what I'm going to do is talk through various aspects of PyMOL and things you can do with PyMOL. I'm going to give you a tour of the interface and a little bit about how to use the mouse. So a little bit about colors, how to color molecules, a little bit about selections. I'll point you to a good resource on selection algebra. I'll talk about how you represent molecules, labels, how you can build measurement objects. I'll try and show you some mutagenesis. I'll show you a little bit about crystal packing and how you can generate the crystallographic images in a crystal structure. I'll try and touch on movies as well, but I'm also very open to questions as we go. So one thing that I'm going to also use during the talk is the Protein Data Bank, and they have a fantastic resource at PDB 101, which if you have never come across, pdb101.rcsb.org contains a wealth of educational materials about proteins and biomacromolecules in general. So we can also take a look at this and download a couple of structures as well to highlight various aspects of PyMOL. I'm hoping that everyone has installed PyMOL. I ended up using a method which wasn't in the instructions called Homebrew to install PyMOL. I was having trouble with Mac ports recently. So I'm just going to start PyMOL from the command line like this. So I'll just show you, actually, let me just do one little thing here. Let's go to, I'll show you what I did, which was Homebrew. Search for Homebrew. And Mac OS. I'm on a Mac, as you can probably tell. It's called the Missing Package Manager. And the way to install Homebrew is very simple. You just issue that command there. And then for PyMOL, install PyMOL. If we go to installing PyMOL, we'll put Mac OS in there as well in the title. So that takes us to the wonderful website that I strongly recommend for PyMOL information called pymolwiki.org. And you can see we have under here the open source PyMOL. So a lot of people install PyMOL from Schrodinger's website. And unfortunately, the version that you end up getting is somewhat hobbled. One of the reasons that it's less useful than the open source ways of installing it is that it does not support It does not support plugins, which can be very useful. So for Homebrew, which they say is the recommended way of installing it, you say brew install brew/bio/PyMOL, and then in the terminal that allows you to type PyMOL. And if I just show you where it is by typing which PyMOL, you'll see that Homebrew installs everything as if it were, I think there's symbolic links, as if it were under user local bin PyMOL. The other method that I've used a lot is macports, which requires using sudo. And then there are a couple of extra things that you might want to do before using that. Okay, so enough of that. Okay, I hope you're not afraid of the command line. We're going to be doing some typing. And this is what we get when it starts up. You can see if I scroll back in this upper part of the window, I've got version 2.4. It was originally written by a wonderful developer, Warren Delano, who I knew, absolutely lovely guy, and he firmly believed in open source software, and I'm very happy to say that it continues to be distributed in an open source fashion and can be installed using open source methods. I should also make clear that I don't work for Schrödinger. I'm an academic at University of Oxford. So okay, so we've got our PyMOL. So let's have a little look at this interface. So we can see that we've got. Let me just move this down out of the way. Are we getting
Garrett,
any questions at all, Chris?
Not at the moment, but could you perhaps make the PyMOL window a bit bigger?
Yeah, of course. So I'll do that by I use I can't really do it any other way than that. Is that any better?
Yeah, I think people. It's just that writing's a bit small in some of the things, but yeah, that should be fine. Yeah.
Yeah, I think I can do that as well. Does that help?
Yeah, a little bit.
Because I can artificially zoom in. I'm not sure if you're seeing it in the resulting. Do you see that? Is that zoomed up? Hello, Chris?
No, I don't think so. No, I think we just.
Okay, it's just a special effect on my desktop, I'm afraid. Okay, so all right, I'm afraid this is what we're dealing with. One thing I could try is to change the screen resolution. Let's see, where are we? Displays. Try it like that. Is that better?
Yeah, that's better.
Okay, all right. So okay, I've made it full screen. So we have now in the upper window a command line, which is indicated by that PyMOL greater than, and we have a set of buttons up here. The builder, which I'll take you through briefly, where we can build either organic molecules or peptides, even proteins, and then a couple of other buttons that are useful for viewing things or setting up movies. And then on the sidebar here, we see the list of all the objects. In the bottom, what the mouse is set to do, and I don't know if you can see in the camera, but I have a 3-button mouse. The middle button is also a scroll wheel, which is terribly useful with PyMOL. So down here it tells us we've got 3-button editing mode right now, simply because I use the builder. If I click there, it goes back to viewing, which is what you get by default. And then we can see how the modifier keys on the keyboard, so Shift, Control, Control, Shift, etc., single click, double click, affect various things. Then we've got selecting, and if I click here, that changes the level at which we select things. So I can change through to chains, segments, objects, molecules, C alphas, and atoms, and then it goes back to residues. If I hold down the Shift key, I can go backwards, so I can go backwards to atoms, or don't press shift, go forwards to residues, chains. Then we've got some playback buttons down here, which I'll show you. We also have an S, which can show the secondary structure, the sequence, the amino acid sequence of the protein. Okay, so this is pretty boring. We always have a button here for all objects, and then we have A, S, H, L, and C. So A is action, S is show, H is hide, L is label, and C is color. And those are very quick ways of accessing various things. And then we've also got the PyMOL window. So let's start by loading up a protein. And I'm going to load up cytochrome P450 from the protein data bank, and I happen to know the 4-character code PDB ID for this structure. So this is cytochrome P450 camphor from putidorredoxin. And we press enter, and in this version of PyMOL, we automatically see the cartoon representation. There are some earlier versions that show it as, I believe, it may well load up like this. So if I say show as lines, you may actually see it like that when it loads up in the earlier versions. So what I'm going to do now is quickly demo some of the things you can do with the mouse. So currently I'm not doing anything, I'm just dragging it around, I'm not clicking any buttons. If I click and drag, it starts to, when I'm using the left mouse button, it starts to rotate. If I shift drag, I can select. And when things are selected, we get these magenta squares that appear highlighting each atom. And if I just click shift on one atom, it clicks at the level that we're currently selecting at. So residues is the current level. If I wanted to just select an atom, I shift click there, or I can click until I get to atoms. And then if I click on an atom, I just select that one atom. And it tells you up here what's being selected. This is really helpful because it actually shows you something about the syntax of the selection syntax hierarchy. We have the name of the object, the segment ID, the chain ID. So in this case, the PDB ID is 2CPP, the segment ID is A, the chain ID is A, and then we have the residue, which in this case is glue 171. So we have the residue name and the residue index, and then we have the name of the atom that I clicked on, which is C gamma in the side chain. All right, so let's see if we can make this look a little bit more interesting. So I'm going to go to A action. Actually, no, I'll do show first of all. Show cartoon. And so what that does is it reads the secondary structure records in the PDB file, and then uses various symbols to indicate various geometries to indicate the secondary structure. So you'll see these coils for alpha helices and these arrows for beta strands. And one of the things we can do is actually color the secondary structure differently. So if we go to color by SS, then we can choose on the menu the various ways of coloring things. So if we choose blue helices and magenta sheets and pink loops, it looks like that. So we can now see how the crystallographer assigned the secondary structure. Now sometimes different crystallographers choose different assignments for the same protein, and the definitions for the secondary structure vary. So I find that if you type DSS and then the name of the object, it will you can use the DSSP method to look at the geometry of the hydrogen bonds and so on, and for each residue and the local environment, and it will use the rules of the Chris Sanders DSSP method to correct or use a consistent definition for the secondary structure. You can see that things change quite a bit there. But one advantage of that is that it uses a consistent method across all of the structures that you apply it to. So,
Garrett, this is probably a good point for this question, actually. Is there a way to build missing parts of protein when you get it from PDB in PyMOL?
So there are. It's tricky. It's very involved, and it's probably not the best way of doing it. Probably the best way of doing it using open source methods would be to use Andrei Shali's modeler method. So you can certainly build in amino acids and then connect them and then energy minimize them. It is certainly possible, but it's a bit of a faff. You're better off probably using something like Modeler to do that.
Would it be true then to say that we should regard PyMOL as a viewer?
It has more capabilities than just a molecule viewer. So it's very good at doing. It can do energy minimization and so-called sculpting, and I'll try and demo that at some point during this. But I mean, it is largely a viewer, but you can indeed do energy minimization using its sculpting feature. Okay, so let's. So one thing that I should note is doing undos is quite tricky. It's not terribly good at doing undos. So I rarely use this simply because it doesn't seem to do very much. You can click at various states down here if you want, and you can restore the situation at various points through this undo stack that appears down here in the more recent versions. These are effectively like scenes. So if I go back to this and say color by secondary structure and show cartoon, we've still got the updated DSS secondary structure. So let me show you some other presets. So you can very quickly. Okay, so I guess I should show more about the mouse first. So I've been just clicking on the left mouse button and dragging, and what you need to do is think of, if you like, a circle around the object, around the center of the viewport, and in that circle is a sphere, a ball, a trackball, actually a virtual trackball. And if you click anywhere on that virtual trackball, it's as if you're spinning the object using that. If you click outside of that circle, you're actually changing the Z rotation, so the axis that points towards the camera. If you put the camera the cursor in the middle of that virtual trackball, you're just rotating around the X and the Y axes. So that's the X axis there, and that's the Y axis there. So if I move left, right, I'm moving around the Y axis. If I move up, down, I'm moving around the X axis. If I click in the if I right click on my mouse and drag down, I can zoom up, and if I drag up, I can zoom out. So that's a couple of useful operations. If I click on the middle mouse button, I'm actually translating the object in the scene, or actually I'm actually moving the camera. I'm not moving the object. Subtle difference. And then if you do have a scroll wheel, if you scroll it, you'll notice that the depth queuing changes and also the clipping. And the more I scroll up, the thinner the depth queuing slab becomes. And if I scroll in the opposite direction, the broader or deeper it becomes. And we use a technique called depth queuing, which changes the shade, if you like, of parts of the object that are further away and blend them more into the background. So it's a bit like a fog. So if I were to go back to, say, that representation, and I can change the background color, so I could go to display background, and I can choose some presets. So I could say background white, and now it's set, ready to do. I'm ready to set up an image for publication, for example. For this, I'll just go back to what I had, which was black. You can also type BG underscore color, and then the name of a color if you want. And gray is one of the colors. So you can do it from the command line as well. Okay, so I'll put it back to black. All right, so let me show you some. Let's see, I've told you a little bit about the interface. One last thing that I want to show you is if I press the escape key on my keyboard, I get this. So it's actually, the viewport is modal, and I can alternate between the history of commands that I've typed so far and the actual graphical view. It's exactly the same as if I were scrolling back up here. One nice thing about having both of these windows is that I can actually highlight and select and copy and paste using my operating system in the upper window, whereas it's not possible in this viewport. And if I type something down here, oops, type in the viewport PyMOL command line, it doesn't appear up here. Okay, so that's a little bit about the interface and the mouse. Okay, so let's show you a little bit about colors. So right now we have carbons as green, and we're using CPK coloring for everything else. So we're coloring oxygens red, and you can see these little what look like asterisks or 3D plus signs. Those are the oxygens in water molecules. We can see blue atoms; those are nitrogens. You can see some in the center there, in the tetrapyrrole of the heme. I'm looking there edge on. You can see the red oxygens of the propionate groups of the heme. We can also see in the middle there the camphor. So I can change the coloring if I click on the C and color by element. And then the very first one, you'll notice there's not even a letter C in that, and that's quite useful. For instance, let's see, if I color the entire object, I can go down to these color groups down here, and these are the names of the various commands, the various colors, sorry, that you can type on the command line. So if you wanted a background color that was 80% gray, I could type gray 80. But I'm going to color the object. Well, let's color the object, I don't know, wheat. Okay, so now I've lost the CPK coloring on the atoms. What I need to do is go to the color by element. If I choose the first one, it remembers what it doesn't touch the colors of the carbon atoms. So now I've got my color by element and my preferred color for the carbons. We can also go through and there's various sets here already. So if I wanted, say, carbons to be, I think that's pink, I'm not sure. Yeah, I could use that one. I could go down into other sets, and you can see the color of the carbon there. So the only difference is that the carbons are changing color each time. This that last one, you got some wackiness on what the way the hydrogens are colored. Oops. Where are we? Yeah, so that's variation in coloring the hydrogens, and analogously, that very last one has, well, that very last one has carbons. Sorry, hydrogens is black. All right, so I'm going to go with white carbons. So that's a quick intro on the colors. You can also color by chain. So if you've got multiple chains present, you can use one of these. So that we've only got one chain present here, and I can also use chain bows, where this colors the sequence from the N to the C terminus. And so we can see also this sequence. So I can either click this S down here, and that will show the sequence, or I can go up to display sequence, and that shows the amino acid sequence. So you can see there are some gray atoms that do not have coordinates, but are part of the protein. So we don't even the crystallographer didn't even model those in. And then we've got going from dark blue to the colors of the rainbow, all the way through to red at the C terminus, the sequence of the protein. And this representation, we're seeing amino acids with single letter codes, and any cofactors or ligands have 3-letter names. So if I click on that, it highlights the heme. So one thing that is always useful when you're trying to look at structures is to set the depth queuing appropriately. So if we've got the heme and the cam, say we want to focus on those as being part of the active site. So I've now got a selection. Every time you click on something, PyMOL creates this temporary object called a SALI, or selection. And I can now zoom on that object, on that selection. What that's done is it set the depth queuing to hide things that are too far away from that selection. And it's also set the front clipping plane, so that everything in front of it is not being drawn. So it's as if we've taken a slice through the protein and we're looking right inside the object at the selection of interest. And then we've got all the waters down here as well. So we can zoom back out. So if I click on A, zoom for next to all, we've got everything back in view. So let's show some presets. So these are ways of getting very useful, quick looks at various aspects of the structure. So if we look at simple, what that does is it draws the backbone using what's called a ribbon in PyMOL parlance, which is just a single line from each C alpha to the next C alpha. And it highlights the ligands and cofactors and key residues using what's called sticks. If we now go to preset simple no solvent, this will hide all of the little red dots, which are red crosshairs. Those are the solvents. So that's probably one of the simplest views you can get. If we go to preset technical, that's really messy. We get lots of hydrogen bonds that are present in the structure. I find that terribly unhelpful. Too much information. So we can delete the hydrogen bonds it created. And if we go to a different preset, ligand sites is quite useful. So if we do ligand sites cartoon, it's also helpfully zoomed in. And what it's done is it's selected all of the cofactors and ligands, and it's drawn polar interactions as yellow dotted hydrogen bonds in this case. The polar interactions are really hydrogen bonds. So anything to the waters or polar atoms are highlighted. And those are all within the vicinity of the site of interest. We can also look at preset ligand sites, and we can do a transparent surface. So what this will do is it will calculate the molecular surface around the binding pocket.
Garrett, a quick comment. As you have it at the moment, you have hydrogen bonds going from the heavy atoms rather than to the hydrogens.
Correct.
One of the questions was, is the hydrogen addition in PyMOL reliable? I presume it means for potentially ionizable groups.
Not quite sure what that means. Reliable. What do they? Can you
Based on the pKa, I presume.
So it's quite a simplistic. Algorithm, and it does not do any titration on the protein. If you'd like to do something like that, then you might want to consider, I don't know, something like perhaps reduce or PDB to PQR, or maybe a commercial tool, MOL, for example.
Yeah.
So, yeah, I mean, if you've got unusual protonation states of side chains, say a histidine that can vary quite a bit, then it's not going to be able to solve that for you. You want to use a different tool.
Yeah, fine. That's good.
So there is set H bond set, sorry, H underscore H bond cutoff underscore center, and then you can give a value for the threshold at which you want hydrogen bonds to be detected. So if you wanted to only detect strong hydrogen bonds, then you might want to use a smaller value, say 2.8 angstroms. And if you wanted to detect weak and strong hydrogen bonds, you might want to say change the set off the H bond, sorry, I've done that wrong. I want that to be H underscore bond. You might want to set the H bond cutoff center to a larger number, like 3.2 angstroms, for example. So yeah, there are some parameters you can change. For anyone who is curious, if you go to the setting menu and go to edit all, it brings up a panel where you can type, you can look at all of the settings that PyMOL supports, and you can also do a filter. So if you type in H underscore bond in the filter dialog box, you get access to all of the parameters that PyMOL uses to detect hydrogen bonds. So maybe that might be useful. But it does not change the protonation state of the side chains.
Another quick question for you. Is it possible to view the commands associated with actions, like when you select a preset, for example?
That's a good question. Without inspecting the source code, I don't think it's necessarily very obvious.
So it doesn't record menu clicks and things like this then?
So, I mean, the best you can, it doesn't record the sequence of operations that you're performing. What you can do, though, of course, is save the session. So what that will do is it will save the current view, the objects that are present and how they're displayed, the colors and so on. So you can either save session or save session as. So if I were to do that, I could type in, I don't know, 2cpp.pse, and that will save this current view. And one thing that I would encourage people to do, given that PyMOL has suboptimal undo, if any, in the open source version, is to continually save PyMOL sessions. It may be a bit of a pain, but if you do something wrong or something bad happens, you have a way of going back. So currently, I've got a nice transparent surface here, which allows me to see inside the binding pocket. In this protein, the binding pocket is completely sequestered. So from a scientific point of view, one question that arises is how on earth does the substrate get into the binding pocket? And turns out that there is a substrate access channel. But what we've got now in this viewer is a surface. I didn't choose let's see, preset ligand sites transparent better. That's so if I choose that, it's going to increase the surface quality. And if you look carefully, you'll notice, for instance, up there we have a smoother contour. There are now many more triangles, plus the boundary between the colored patches is now a little bit smoother and more detailed. So if you were going to create a figure for a journal, you might want to increase the surface quality. So that's fine with the presets. If you want to do it manually, again, if you go to setting, and in this case, we're going to type in surface, and we're actually looking for quality. So if you were to change surface quality here, you will increase the surface smoothness. So a good value for publication would be 2. I mean, if you've got a faster computer with more memory, you might want to go 3, but that's really pushing it. So you can do it. You can change it here in actually in the viewer. Sorry, in the GUI, or you can do it here. So set space surface. Oops. Surface quality, comma, and we'll set it to 0. So another thing that is kind of useful when you're looking at a protein for the first time is to just get a sense of the shape of the protein. And
we have a quick question.
Yep.
How do you copy a command? This is under Linux. Control C is not working.
You're saying from this window? Yeah. Or
perhaps the one above. I'm not sure.
Yeah, I'm not. Yeah. So I mean, normally on Linux it's Control C, but you might need to in a terminal use Shift Control V instead of just Control V. The reason being that the Control C in the terminal actually interrupts the process. So in the terminal, you normally have to use Shift Control C and Shift Control V.
Okay, that sounds great.
Hopefully that makes sense. So I was just going to show you how I normally look at the shape of a protein. So one thing you can do is you can go into setting, and here you've got all kinds of extra things you can do. So I'm going to go to surface. I'm going to go to surface color. So it's setting, surface color, white. Okay. Now what that will do is it overrides everything, and now it doesn't matter what you color the object here. The surface stays white. So if I now go to show surface, it's going away, it's computing it, beach ball of death. And I've got a very high quality surface now showing with transparency. So if I go to transparency surface, go off, hopefully that will override the preset. If not, we're going to reload it. It looks like it's still it's inherited the. Okay, so I'm just gonna quit and restart because the presets mess around a lot with the settings. So if we go PyMOL, actually I'll cd to my directory first. So I'm going to go to desktop and PyMOL, and I'm going to start PyMOL. And there we are, we're back. And then I'm just going to say fetch. Actually, I can show you what happens if we. I won't. I think the PSE probably incorporated the settings from the preset. So I'm just going to start afresh. Fetch to CPP. Oops, sorry. That I did not expect. Apparently there's a 2pp. Delete object. Okay, all right. So I'm going to show the surface. And this is what would happen if we don't change the surface setting color to white. It's messy. It's messy. And then if you look carefully at that region there, I've got, you can see the triangle. So there's a flat edge there and a flat edge there. So if I go to setting and surface and color and white. We've now got a very clear impression of the shape of the surface of the object. The waters are currently showing up as little crosshairs. Now, if we do a show non-bound spheres, so now we're actually seeing all of the waters as tiny little red dots. So we can see where all the waters in the crystal structure are sitting. The colors are still there originally. So this is one nice thing about that method of setting the surface color. So that's useful. And then if we want to go back, we can say default atomic. So here we can color the entire thing one color. And then we can do color by element if we want. And now you can see everything in its horrible, gory detail. So I think very often when you're creating molecular visualizations, you want to think about what is the message you're trying to convey. So okay, so let's turn off the surface. So we say hide surface, and I'm gonna set the depth queuing automatically by doing zoom, and now everything is visible. So let's select the camphor in the active site, and we're going to go and zoom on that selection. And now if I click, I'm going to show you a different way of doing that. So zoom on all. So I can also type zoom Sally. Okay, and then I'm back to zooming closely on my object I've selected. If I click on something else, it's adding into the selection, and now we've got the heme selected as well, and altogether it says 54 atoms. So if what I'm going to do now is I'm going to rename that selection because it might be useful. So I'm going to call that active site. So I'm going to press the backspace, the delete key on my keyboard, and I'm going to type in active site heme and camphor. A bit long, but there it is. It's too long. Okay, so I want to see all of the whole name of that thing there, active site heme, etc. If I click on that tiny little sliver there and drag it out, you can see I can change the width of the sidebar. That's really handy if you've got long names. All right, so now how can I find all of the residues that are nearby? So I'm going to click on A. Actually, I'll just click off the background and I'll start again. So I'm going to do a Sally. Okay, so it's remembered the set that I just created, the selection set, and now I'm going to expand this new Sally. I'm going to go to A, and then I'm going to use the modify command. So I'm modifying the selection. I'm going to select around. And I can either choose atoms or residues within some distance cutoff. So if I choose modify around and then residues within 4 angstroms, it's now selected all of the residues that are within 4 angstroms of any atom in the camphor or the heme. And you'll notice that the original things I selected have gone. They're not in that selection anymore. So now I've only got the residues. Not the heme and not the camphor. So if I now show them, see what they are, you can show them as lines. That's one representation. I can show them as sticks. That's another representation. I could also show them as spheres. So this draws the van der Waals radial spheres around everything. And then you get a true sense of the amount of space that each atom or side chain takes up. I could also show them as dots. So I have to hide things now. So I'm going to hide the spheres. And you can still see this, the sticks are still there. But now I'm actually showing dots around each of them. And if I zoom out, you can kind of see it slightly better where the dots are. Okay, again, this is a useful selection, so I'm going to save it. So I'm going to rename that selection, and I'm going to call this active site. I can either do Shift minus on my keyboard to get the underscore, or if you hit space, PyMOL is helpful and it just puts an underscore for you automatically, so that can be useful if you're typing quickly. Active site. Okay, now I can click in the background, so in the black background, and I've lost my selection. The selection is turned, well, not lost, turned off. So again, if I zoom back out, you can kind of see that the dots get closer together. All right. So what else can we do? We can look at polar interactions. So if I click, again, I'm on residue level on the camphor, I can now try and find all the polar contacts that makes with everything in its vicinity. So if I go to action, let's see, I want to find this time polar contacts. This is a new, down here is pi interactions. That's a new thing that's been added in more recent versions of PyMOL, which is nice. Polar contacts, and I'm going to say to other atoms in the object. And it's found one polar contact. And it turns out that's to this tyrosine over here. And it's drawn it by default with a yellow dotted line. If I don't like that, I can change it. So I can go to color, and let's see, let's go to a light blue. So there we go, we've got our light blue polar contact. And I can rename it if I want. So I don't know, let's call this cam underscore HB, and that is our hydrogen bond. So I'm gonna hide the dots for now. Now, you might want to label things for your representation. So I'm going to clip a little bit into the pocket so it's a little clearer. And I'm going to right click on an atom in that residue, that tyrosine. And residue, I can choose the level I want to choose. And then I can do label. And now I've got a lot of options. So I can label the residue one letter. I can do the residue identifier if I want. So let's try residue one letter. So it's created a label at the C alpha there. So that's Y96, and that is quite small. It is chosen white as the color, and it's if I change the background color, so if we were creating an image for publication, we might want to choose white. And now it's changed the color automatically of the label automatically to black. Now, it is possible to edit the labels and move them around, but I don't want to do that while the atoms are still showing, because if I try and drag things around, it'll move the atoms as well. So what I need to do is to hide the sticks on that. So I'm just going to hide sticks on my active site. I'm also going to hide the lines. And then I'm also going to hide the cartoon. We're not going to lose anything. We can easily bring that back. So there's our little label. I'm going to click editing. I'm going to try and command click on it. You'll notice I'm dragging it around. So I'm now holding down the command key on the Mac and the left mouse button, and I can actually move it. And then if I bring back the other bits that I had that I need, and if I say show, let's see, I want to show cartoon. Okay, so it's starting to look good again. All right, so I've got my tyrosine 96. I can also, there's an, let me see if I can just find it. I think it's, cartoon side chain helper. So currently, you'll notice that the alpha carbon of the tyrosine is connected as well as, and is showing the backbone atoms of the tyrosine 96. So we can see the backbone NH, and the carbonyl. And let's see if I do set space cartoon underscore side underscore chain underscore helper, comma 1. Now it's actually cleaned things up a lot. So it's no longer showing the backbone atoms other than the C alpha, and it's also connecting the bond from the C beta directly onto the cartoon. So that's why it's helping. And you'll notice that it clarifies the image a lot. So now, are we good to go? Not quite. So in the heme, there are actually 4 bonds to the iron atom. So if I okay, I'm still on editing mode. That's dangerous. Click out of that. So if you come down to the bottom right and click on viewing again, I'm going to change the level to atoms. So I'm going to shift click on residues and click on the atom. And you'll notice up here it says Fe, the iron ion in the heme of cytochrome P450. So this enzyme, for those who are curious, is a monooxygenase, and it normally binds dioxygen to the 6th axial position of the iron ion. And it does it, it orients it in such a way that the dioxygen points towards this so-called dioxygen binding pocket. There's actually a slight unwinding of this proximal alpha helix just here, enough to create a little divot for the dioxygen to bind towards. But what I want to do is create some bonds here. So there should be 4 covalent bonds to this iron ion. And for some reason, in the PDV file, there's only 1. So what I'm going to do is, again, switch back to editing mode. I'm going to click, and you'll notice I get this sort of unusual looking sphere around the atom. And if I click again, you'll notice that this now has sort of 2 rings in each of the 3 dimensions. So there's just 1 ring there, there's 2 here, could keep on going. So pick 1 is just 1 ring, and pick 2, PK2 is 2 rings. If I type in here at the command line bond, it will create a bond. So now if I click in the background and repeat that process, so I'm going to go from the iron to the other nitrogen in the tetrapyrrole, bond that, and then click there and there. I'm pressing the up arrow in this dialog line to recall previous commands. I can keep going back like that, as you can see, up and down with the up and down arrow. So bond, okay, and then click on the background, and then come back out of 3-button editing to viewing. There is you can also do it, you can change that mode up here in the mouse menu. So motions, viewing, editing, and so on. You can even move around the lights. So okay, so now we've kind of got a view that looks vaguely all right. We've got some things that are appearing in the front clipping plane, which is not always so good. So what you can do is go back to, let's see, residues. And we can just click on those things that are being clipped in the front plane here. And just hide those. So I'm just going to rename that selection something like to be clipped. Oops, you don't want a minus in there. And then the other thing I can show you here, by the way, is if I don't like the order of these objects that have appeared in the sidebar, if I use the right mouse button and drag, I can change the order. So you can set the order up to be what you want. Okay, so now what I'm going to do is hide everything in the to be clipped bit, and so those are gone. All right. So next up is ray tracing. So what I'm going to do is type in ray, and you can either just type ray.
We have a quick question that might be useful here.
Yeah.
Can we display hydrophobic contacts?
So yes, you could if you want. Of course, you have to decide what is a hydrophobic contact. Probably the best way, and it's manual. It's not automatic. So you have to choose. Is to use. I would use perhaps the measurement tool. So if you go to the wizard menu and go to measurement, right? You can now, if you click on distances, you can change to other things if you want. You can choose to measure angles, dihedrals, and so on. So there are heavy neighbors. So that's actually new. Distances gives you the most control. So if we stick with distances, where it says create new object, every time I click on two atoms, it will create a new distance, a new measurement. I could either so it's by default on this setting, create new object. I could either have it to merge with the previous, so I can just keep on clicking. That might be useful if I want to show all the Van der Waals contacts perhaps. So let's use merge with previous, and let's see if we can find what we think are hydrophobic contacts here. So I'm just, I mean, maybe 5 angstroms is probably a reasonable cutoff. So I'm just doing this manually. You can also build a selection using the technique I just showed you about selecting atoms within a certain distance. Okay, so let's just say that those are the hydrophobic contacts we want to show. So we're deciding that, which is not great. I'll mention a way of doing this, a couple of ways of doing this sort of better than that. So let's say we're done. So that object is called measure01. Let's rename it to something more meaningful. Oops, sorry, I hit reset. Rename. We could call that hydrophobic contacts. And again, we can color using some appropriate value. Let's choose TV orange. It's currently showing the distances. We probably don't want those. We might not want those. So we can go to the H and hide labels. And so we could do it that way. But really, probably what you would like to do is, sorry, let me come out of full screen. I'll just show you a tool that is open source and might be useful. It's called Arpeggio, and it comes from Tom Blundell et al. So there's the paper, and it can generate PSE files for PyMOL that show all of the different kinds of interactions that it can detect. So it can go away if you give it the appropriate input. So the web server will do this as well. You can install it and run it on the command line. It's probably preferable. So it will look at polar contacts, hydrogen bonds, hydrophobic contacts, and you can download the PyMOL session and then load it up. So that's probably the best way of doing it, I think. Okay, so let's come back to this. So we're turn off the hydrophobic contacts. So let's say we're happy with this. Probably want to hide another. Let's go with that. Okay. So now I was just about to show you ray tracing. So if we hit ray, what it will do is it will computationally cast a virtual ray across the scene, pixel by pixel. It's interesting, Siri woke up. And
okay, I found this on the web.
Sorry about that. And so it casts a virtual ray throughout the scene, pixel by pixel, and decides whether it's in shadow or not, whether it's the edge of an object or not. And what it does is so you'll notice there are now shadows, for instance, in this scene. You can see shadows there. But it also gets rid of aliasing. So aliasing is a molecular or computer graphics term that says that if I were to try and draw a diagonal line using pixels, it would go up step by step in a staircase effect. The way anti-aliasing works is it puts a gray value instead of a black or a white, and it blends into the background. The other thing you'll notice is that the background here has changed to a checkerboard pattern. So now by default, PyMOL, if the background is white, I think it does it with black as well, is it will blend to a transparent background. So we have a red, green, blue, red RGB and alpha transparent channel. And so now what we could do is save this view. So we say file, export image as PNG. And we're going to do the full image, and then we click on Save PNG As, and then that gives us a chance to type in the name of the file, so 2cpp active site, and it would be a PNG. If we save it, we've now got the image saved. If you don't like the transparent background, which in most cases is useful, you can, where is it? Background, is it? Opaque, yeah. So display background opaque. So if we were to do the ray tracing again, it should just be a white background this time. So yeah, we can, that's optional there. All right. Okay, and then the other thing you can do with ray tracing is you can give it the pixel dimension. So if a journal insists on a certain size, you can also do that. So I don't know, let's make it really small. I don't know, 480 by 360, I think. Is that right? Can't remember. What my syntax? Sorry. Is it that? I'm gonna have to look it up. Yeah, it is comma. Yeah. So we've now got what we were looking at, but now squished into a 480 by 360 viewport. So and then when you click in the window, it goes back to the original view. So I think that's sort of a quick intro, relatively quick intro to visualizing molecules, a little bit about colors. So I did mention there's selection algebra that's worth knowing about. So if we go to the PyMOL, so if we do, I think it's PyMOL wiki. Selection algebra. Oops. And then that takes us to the selection algebra page, which is terribly useful. So we could, for example, combine the solvent set and chain A. So we could now show spheres just for the solvent in chain A. So the selection operator modifier table lists all the atoms. So you've got all the operators and any aliases that might exist. Logical operations. So you can use not, and, or, implicit or. You can even use first and last. There are a whole bunch of useful identifiers that allow you to access different hierarchies in the structures that are loaded. So chain is very useful. Res n for residue name. So res and allo would select all of the alanines that are present. Res-I is the residue identifier, which you can see when you look at the sequence. Name is the atom name, so Ca would select all of the alpha carbons. If you have any disorder in your structure, you might want to select all of the alternate locator atoms that are labeled A with Alt, and then the A, or the desired alternate location indicator. There are also, you can also access things using the index, which is how PyMOL reads things in, or the ID, which is what's in the PDB file. You can also look at things that are in a set. We can do things at various levels. So I showed you this, the change in the selection level. So by object, by SEGI, by segments, by chain, etc. You can do bonds, proximity, which I kind of showed you with the modify command. So around or expand, near to, those are useful. You can do things with properties. So if you have partial charges in the object, you could look at that, or anything with a B factor less than or greater than some threshold. There's a bunch of useful flags. There's some shorthands for organic. That's really useful for finding ligands in the molecule. Side chains can be useful. For example, you can hide all the hydrogens, hydrogen bond donors and acceptors. You can look at things of this style. You can also again use qualifiers like near to, beyond, around, and so on. And then you can also use parentheses. So chain A and not residue 125, for example. So lots of useful examples there as well. So and then yeah, you might also want to look at selection macros as well. So this is a really useful web page to bookmark if you or search for. So PyMOL wiki selection algebra. Okay, so let's take a look at. I've touched a little bit on measurements. We could also look at mutagenesis. So in this example, what might happen? So let me bring back the things that I clipped as sticks. So they're still there. So let's see what might happen if I were to mutate my tyrosine 96. Actually, we'll leave that. Let's take a look at this guy here. So this is V87. So let's try mutagenesis on that. So let's see, can I mutate in a tryptophan? So I can go to mutagenesis, and we want protein because we want to vary amino acids. It brings up this little panel down here. By default, it says no mutation. So that can be useful if you want to explore rotamers of side chains without any change. So if I just click on the residue we selected, Phe87, you'll notice now that I've got the original and the mutation. And I'm going to color that mutation slightly differently just to bring it out so you can see it. Let's do it in pink. And there it is. So our mutation is currently in pink. The original coordinates are in white. And you can see some cylinders that have appeared, pancake-like objects that have appeared that indicate the steric clash for this current rotamer. You'll notice also here a percentage, and that is the average population of that particular rotamer in our normal, in the set of structures that exist for possible structures that are available to that amino acid, so in this case, Phe. So about 10% of the time it'll be in this rotamer. I can now explore other rotamers. It's saying 2 of 4. So it's telling me that this is the second most populated rotamer in for female alanines. If I go and if I hit the left arrow, I can go to the first rotamer, which is most commonly observed, 82% or almost 83% of the time. You can see now that I've got a lot more red clashes, and it's clashing strongly with the adjacent tyrosine 96. So that's not a terribly good rotamer. I don't think I'm going to go for that. If I go the other direction to 3 of 4, that's really bad. So that's clashing with the alpha helix that it's adjacent to. That's no good. And then the last one is clashing with atoms that are not shown, possibly the hydrogen that's attached to that water and things over here as well. So that can be useful just to explore other rotamers that might exist for the side chain that we currently have. But now if I choose no mutation and tryptophan, okay, now it's automatically selected the one that fits the best, and it's still not great. There's some nasty clashes up here, and if I step through the others, you'll see that they are not happy. So that is never going to work. I probably wouldn't want to ask my molecular biology friends to go away and make that sequence. Okay, well that's mutagenesis for you. The dotted line is an affordance that PyMOL puts in when it has a gap in the sequence. So that is not great. And I apologize for that. So, okay. I don't know why that happened.
One of the questions we've had is, would it be possible to discuss how to work with molecular dynamics trajectory in PyMOL?
It's not really designed for that. I mean, you probably would want to use a tool like VMD. So let me see. One thing I can show you. Got an NMR structure with about 100 odd. Let's see. Yeah, 2K39. So it's a ubiquitin. Okay, so let me see. I'm just going to reinitialize everything, and I'm going to fetch 2K39. Okay, so this is a ubiquitin. So if we just go to the PDB, find out all about it. So 2K39. Oops, sorry, that's not the PDB. Or CSB. 2K39. So we've got the macromolecule entry ID 1 ubiquitin. There it is. So if we go back in here, again, I can color it by rainbow. Actually, color the chain by chainbows. So now we've got the entire chain. So it's a lot smaller than the previous protein we were looking at. But you'll notice down here the state is 1 to 116. So I think this is a really nice example of protein motion. So you can indeed certainly visualize multiple states and you can actually export this as a movie as well. So I mean, it's not really designed to visualize trajectories. I would use something more like VMD if you're interested in MD, molecular dynamics. Okay, so let me show you how to align structures. So what I'm going to do, stop that. Oh, just FYI, we've got tons of states here. If I want these as separate objects, there's a command. I hope it's loaded in this version of PyMOL. Split states. I have to give it the argument as well. So 2k. Okay, that has now created 116 different objects here, and they're all being displayed separately at the same time. So you can see there's a huge variation in the N-terminus. And if for some reason you want to see those separately, you can turn on something called grid mode. And now I've got every single state that's loaded. It's now a separate object in its own little viewport. And if I drag, you can see the conformations are changing at the same time. So that's an extreme example of grid mode. Okay, so let's reinitialize. And I'm going to load a couple of HIV protease structures. So I'm going to say fetch. That's going to go to the PDB and look for these PDB entries. So I'm going to get 2NXM. And let's try 4OBK and see what happens. Okay, so we've now got 2 HIV proteases present. And just to show you grid mode in this case, so they're not terribly useful in those orientations. So what I'm going to do is show you how to align them. I'll go back out of grid mode. And the simplest way is to use the align command. So let's try aligning the second one, so 4OBK that I loaded onto the first one to NXM. So so I did align to molecule. You can also work on selection. So I could, if I had a selection, I could go to a selection. And there we are. So now if I zoom on the original object, I can also use orient. And what that does is it looks for the major axes of the object and puts those major axes along the main axes of the screen. So now we've got our 2 HIV proteases loaded. And it turns out that these are actually not quite HIV protease because they're mutants. And they've been muted to be catalytically inactive. So if I show the secondary structure, sorry, I keep saying secondary, I mean primary structure, we can see here the sequence of the amino acids. And normally at position 25, there is an aspartic acid. So you'll notice in these sequences, it's actually an asparagine. So I'm going to rename that D25N mutants. And if we show them as sticks and then zoom, you can see that those are indeed asparagines. And normally we have aspartic acids. So HIV protease is normally an aspartyl protease. And the cool thing about these structures is that we're actually seeing substrate in the active site. So if I change the selection level to chains, I choose, you'll see up here, first of all, it highlights in this scroll bar the bit that I've selected, and that can be useful because if I drag the scroll bar over that bit is now visible in the window. You'll notice also that the color is gray there. That's telling me that the alignment didn't work very well there. There wasn't a very good RMSD across those regions. If I turn off the ALN object, it goes back to the original coloring. Okay, so I'm going to select the other chain, and then those are the substrates. So I could say, let's see, show those as sticks, and you'll notice that I've now got two different substrates showing in the active site. So I'm gonna just color those white with white carbons, and I'll zoom on those. I'll rename those actually to substrates, and then let's zoom on those. So what PyMOL has done is use the protein backbone, the protein structure, to do the alignment. And that's what the substrates look like when bound to those HIV proteases. So you can see that the conformation is fairly extended, and they occupy similar regions. Let's just double check. So yeah, so the 2NXM is the cleavage site, or peptide representing the cleavage site between the RT and RH, and the 4-OBK is between the P1 and the P6 in the HIV polyprotein. So if we choose that chain, then again we can do show. A. We want find polar contacts to other atoms in the object. It's actually created another object over here, which doesn't behave well in this grid mode. So this is where groups come in handy. So what I'm going to do is, so I created that for the 2NXM. So I'm going to rename that 2NXM space, we call it HB for hydrogen bonds, and turn that off. Do the same thing again. So A, we want to find polar contacts to other atoms in the object. I've got another one, and that's for the 4-OBK. So OBK underscore HB. I'll turn that off. So now what I'm going to do is I'm going to create some groups. So if I say group 2NXM underscore GRP, comma, and then 2NXM plus, which is the same as or, 2NXM underscore HB, I created a group. And you'll notice that the 2NXM that was up here has gone away. It's actually down here in this group. If I click the plus, you can see that the objects I asked it to group together are now indented and in that group. If I do the same for the other one, so it's going to be group 4-OBK, and I need to give it a name different from anything else, so GRP, comma, and then 4-OBK plus, and then we want 4-OBK underscore HB. We hit enter, and now we've got two groups. And now if I try and turn on the grid mode, and I enable those objects, they're now present. They're no longer occupying separate panels in this view. And I could if I want, change the colors of the distance or polar contacts if I wanted to match. I can perform operations on all the objects at the same time. So if I want, I could say all show non-bound spheres, and there we go. And I could hide all of the lines, and then all of the little crosshairs have gone as well. Okay, so that's a line. So I should also show you this. You'll notice that the cursor changes when I hover over the boundary between the viewport and the upper control area. If I click and drag, I can actually change the relative size of the parts of the interface in this particular version that I have. You may find if you've got the TickleTK implementation, this upper panel is a separate window, and then the viewport is in a second window. Let's see. I wanted to show you help. So if you're ever curious, you can just type help if you don't know how a command works. So help, and then these are all the things that it can help you with. We could say help align, and then it will tell you if it has a help page for you, all about that command. So help align says, align performs a sequence alignment followed by a structural superposition and then carries out 0 or more cycles of refinement in order to reject structural outliers found during the fit. Align does a good job on proteins with decent sequence similarity greater than 30%. For comparing proteins with lower sequence identity, the super and CE align commands perform better. So I showed you the simplest method, align, which has an analogous option in the menus. So you could, so normally it's align mobile target. You can say super mobile target. Chris, is there a question?
Yes, a couple of people have asked, is it possible to visualize docking results?
Yeah, sure, it is. So you can actually in PyMOL load up PDBQT files. They're close enough to PDB files that they'll behave like PDB files. You can certainly load up SDF files and play through them using the play button down here or the arrow keys on your keyboard and go through them one at a time. And again, use these tools to find interactions and so on. Yeah, so.
Yeah, a related question, and I think you probably answered it already, is once you've got a visualization and a session that you're happy with, can you then read in a docking results? And I think just reading in the SD file would do that, wouldn't it?
Yeah, totally. Yeah. I mean, I know I've got tons of docking results on here, but finding them, I don't want to troll through my system. But yes, you can certainly do that. Yeah, and save the session, and then it remembers where everything is, and so on. It may not. I mean, depending on what kind of docking tool you used, you may lose some information, or it's not easy necessarily to see the scores. There might be ways of getting that through into PyMOL. You could change the B factor column in the PDB format to be the, I don't know, the score of interest, and do things there.
Can you remove solvent molecules when the solvent is not water?
Yeah, if it's a consistent residue name, yeah. So I think it's the remove command. So if we say help remove, so you would do remove and then remove, and then. So for waters, I would say resin HOH, residue name HOH, and then if I do that here, it blasts away all the waters. But you could equally say, I don't know, resin, I don't know, is it DMS for DMSO? I don't know. Yeah.
I mean, many of these solvent molecules have 3-letter codes, don't they, in the PDB?
Yeah.
You just have to learn them, really.
Yeah. You can just look at the file.
Yeah.
Yep.
And going back to your looking at the NMR results, does PyMOL show which conformation is stable?
No. Well, you have to. Well, in this case, that's a trick question in a way. So the NMR data that you get back are a set of distance constraints, right? For that protein in solution, usually at whatever temperature. So all of the states that you're seeing are, in principle, valid conformations that agree with the experimental data from NMR. So you may want, if you were to stick them into a force field and compute their energies, then you may get different answers. But that is the experimental data. Maybe I could show a little bit of sculpting.
Well, but the other thing that I know people have always asked me about, it's probably worth showing, is when you're trying to do something for a potential docking study and you have a protein structure, how do you get rid of the ligand that's already there?
Oh, okay. Yeah. Okay. So imagine we wanted to dock to one of these HIV protease systems. I just turned on rock, by the way. So I'm going to get rid of one of them just for clarity's sake. So I can delete the group first, and then delete. Okay, let's get rid of all the objects. So I'm going to get rid of, let's see, action, delete that one, get rid of the line object, setting this selections are fine. Okay, so say I want to get rid of the peptide here. So what I would do, this could be the ligand, is select it, and then I just remove it. So I could either go to Celi action, remove atoms, or I could just type remove Celi. So now I've gotten rid of I've gotten rid of the peptide that was present. And so if I were to now save that, I would say export files, export molecule, if you want to use the menu. Selection, no. I want to choose that object to NXM. That's the one that's currently enabled. And then I click save. I could look at the PDB options if I want. That's all fine. Click save. And then this would be to NXM underscore no. We'll call it lig. It was a substrate, but no lig. Dot. And then you can choose the format you want to save in. So for docking tools, you might want to use PDB. You might want to use MOL2. It's entirely up to you. Put the 2 dots. So that's how you would get rid of the ligand ready for docking. I do have a script somewhere that it's called makebox that will take an autogrid grid parameter file and turn it into a PDB file that you'll load into any viewer, and it'll show you where the grid box is if you want. That can be useful too. Yeah, are there any other questions?
One question was, how did you get the aligned objects to appear in separate grid spaces?
Okay,
how do you undo it?
Yeah, okay, so I'll show you. What I'll do now is I'll take this is a homodimer. So if I color this by chain, right, you'll notice that I've now got a green chain and a cyan chain. And they're actually part of the same object, okay, which is 2NXM. So what I'm going to do is I'm going to select one of those chains. So I'm currently on selecting chains. I just clicked on any atom here. I can click on the background and I could click on any residue in the sequence, and that selects the chain as well. So I'm going to make that into a separate object. So I'm going to drag with my right mouse button the Celi to the bottom of the list, and then I'm going to go Celi A. I'm going to use the extract to extract object. Okay. So I've now got 2 chains. I am still in grid mode turned on. So the answer to the question is set grid mode equals either 1 or 0. So if it's 0, it's off, and if it's 1, it's on. And they're now in their own separate viewports, which can be very useful for comparing things side by side. So if I go back to 0, if I know, so it's by default created a new object called obj01, which is never very useful. So I could now rename that. I'll give it something like 2nxm underscore b for chain B. And now I've got two separate objects. It's not part of that original group. And if I wanted to do the inverse and create a brand new object from these, what I would do is I would select the two things. Before I did this, I would make sure that the chain IDs are different. So if they're the same, PyMOL gets incredibly confused. So it would end up having 2 different sets of coordinates for residue 1 in chain A, and it creates all kinds of problems. So one thing you might need to do is to alter the chain of the object. So just going to search for I don't know. Let's see, 1H SG, for example. So I know that's an HIV protease. It is a dimer. I don't know. I think of a good example. I don't have a good PDB code to hand. I'll just show you. So let me copy the original chain of original chain A to a new object, and I'll rename that. Call that chain A of 2nxm underscore A. And then I'll turn off the group. And let's imagine that we've created a second image, second job object from let's see. So there are some HIV protease structures where you've just got one chain in the asymmetric unit, and the symmetry is such that when you look down the C2 axis, it would generate the second chain. And if you were to do that, you would end up with 2 chain A's. And if I were to now try and merge these together, it would create all kinds of problems. So what you'd have to do is make sure that you change the chain ID to something different. So I mean, B is a reasonable. That's what it was. And now it's possible to create a new object. So I'm going to click A, copy to object, new. If I turn off the original 2, you'll notice that I've now got an object with 2 chains called obj01. And if I rename it, it's 2nxm underscore AB. So that's how you go the opposite direction. Are there any other questions?
One question which I feel that probably a lot of us struggle with is sometimes I struggle a lot to get a good view of protein. The structure may be very crowded to create a figure. Any general tips on things to do?
Yeah, sure. I mean, I think the example with the cytochrome P450 highlighted it quite well. Let me just see if I've got any other quick examples I might be able to load. Let's see. Yeah, let's go back to the P451. So I can actually load existing files. So if I type load to CPP and hit tab, it's listing all the matching files up here. .cif. If I use the fetch command, it will use the CIF format from the CIF format from the PDB, which is a slightly more recent version of that. So I've got my P450, and let's say I really am only interested in the active site, and I'll get rid of all the stuff around it. I think what you need to do is build up the selection of the region that you're interested in, and then just hide everything else that you're not interested in. It's a very simple principle. We'll rename the selection. Call that heme. Call it heme cam. We'll zoom on that. And then we'll select some residues around there. Actually, I'm going to click off, rebuild the selection so I've got a temporary one I can expand. So A, modify around residues within, say, 4 angstroms. I'll call that active site. And what I'm going to do is I'm going to hide everything. And then I'm going to show sticks for that part. I'm also going to show the cartoon for that part as well. I'm going to turn on set sidechain helper. Make sure I get this right. I'm going to choose. So one piece of advice is to try and minimize the visual noise. So I mean, if we were creating an image for publication, we would probably want to set the background to white, and let's change the carbon atoms also to white. So color by element that. And then we want to bring back obviously the heme. We're currently still green. Color those like that. So it's still, still quite noisy, and it really boils down to what is the question that you're trying to answer, or the message you're trying to convey. And I think if this boils down to lots of manual work, so hiding bits that you don't want to see that confuse matters. So I'm selecting things that are kind of in front of the view. So I can do this. I can shift drag, and then that I'm just going to make a note of what I've selected. I'm going to rename it. And then hide everything there. And so now I've got a view that looks, it's inside the protein. It's close to close to the bits that I'm interested in. If I want to highlight things of interest, then I can start to use contrasting colors. So say I want to highlight the camphor. I might want to choose, let's choose something with a dark color on the carbons. something like that, perhaps. And then if I were to ray trace it, it highlights, it brings attention to the bit that I'm interested in. I mean, it does take, I think, time to get familiar with the protein and the best angles. And there may well be conventions in the literature about how things are depicted as well that you might need to follow.
Okay, we have another question.
Mm-hmm.
Is there a way to include if operator when making selections?
So conditional. So there kind of is with, if we go back to the selection algebra, you've got the where you've got sort of conditionals with distances in things like within, around, expand, and so on. And then you can also do if less than or greater than for properties. So so there is that possibility, or equals, or it's in some set, in some range. So there are some affordances for conditionals in the selection. Yeah, so I think the answer is yes.
Okay.
I mean, you can certainly write your own scripts as well. PyMOL has its own language, PML, and you can also iterate through objects present and operate on them. That can be very useful, very powerful. You probably want to begin by saying something like, I think it's from PyMOL input CMD, I think. I'm guessing here. Old finger memory. Right. And then I could say LBJ, let's say, I don't know, call it all, equals CMD dot get, oh gosh, get all objects, I think, get object. I'm gonna have to look that one up.
This sort of brings in one of the other questions we've just got, which is, I mean, you're talking about the PyMOL programming language, but the PyMOL's written in Python, isn't it? So could you comment on the interaction with Python?
So, I mean, it's written in C originally, and it embeds a Python interpreter. So let's see. So I think if we say import sys, right, and I think it's is it sys.path? Sorry, we want to print it. I think. So that tells me where it's getting PyMOL from, where I could install things. So if I wanted to, you know, sort of interact with my own PyMOL, then I would want to install any packages that I want to access in here in something like user local, the Python 3.9 site packages, for example. So yeah, I mean, I'm pretty sure I don't have, for instance, RDKit installed with this version of Python, but there's nothing in principle stopping me from doing that, and then using RDKit inside PyMOL, for example.
Okay.
Are there any other quick questions?
No, I think that's about it, actually. Yeah,
okay.
You've got lots of compliments on the presentation, which is always nice to see.
Cool.
And yeah.
I did want to show sculpting as well, so I'm going to maybe try and finish with that. So by the way, this is all Qt based, so I can pay, oops, I can rearrange things in the interface. So I can put that builder part up there. So you'll notice there's a button called Sculpt. I'm currently on chemical. Let's go to proteins. I'm gonna try and turn on sculpting through this interface. So you can also do it where is it? Here, sculpting. All kinds of hell might break loose if I do this, but let's try it. So show, I'm going to show cartoon. Ugh, too much. Hide cartoon. Okay, let's show ribbon, and I'm going to color that also everything by element. There we go. Right. So if I turn on sculpting, plug in sculpting. Okay, so it's now running with, so it wants me to pick the center atom, and it's going to do residue shells by default. I'm going to increase the radius of energy minimization to 10 angstroms, and there's a cushion of 6 angstroms, and it wants me to, it's telling me up here, please pick the center atom. So I'm going to click on one of the atoms in the camphor, so that one. And you'll notice the color has changed, and things are slowly moving and relaxing. And so it's actually performing an energy minimization right now, and it's blown up the tetrapyrrole. So that's horrible. That's a really bad mess. That could have been prevented if I'd created the bonds to the iron-ion. Cautionary tale, it's not necessarily parameterized for everything you want to do. The other thing that you can do is you can actually, forget the right mouse combination, move atoms. So I think it's control and the first atom. Let's try that. Control and an atom. No, that's doing torsions. It's not quite what I want. I think it's command. Yeah, command. So I'm now actually dragging the camphor around in the binding pocket. So I'm holding down on my Mac keyboard a command key and the left mouse button. And you'll notice that the protein is relaxing as I create steric clashes and effectively creating molecular mechanical garbage. And so drag things around. I haven't completely destroyed everything, but that tells you that it is entirely possible to create do energy minimization. I can toggle bumps. So with that hopefully turned on, why it's not showing. Okay, I don't know why it's not showing, but it should. I think you have to do that before you turn on the sculpting, but before you start the sculpting. So sculpting, toggle bumps, and then click, I think. And then that should show bad context. I don't know why it's not doing it. Anyway, yes, you can do energy minimization. You can build molecules using fragments, molecular fragments, and so on. I didn't show fixing. You can fix atoms and only energy minimize subsets of things as well. So there is a bit more to PyMOL than meets the eye. Yeah.
Okay.
Okay, good. Thank you.
Well, in fact, as I said the last time, if there was a virtual applause button, I'd be pressing it now. I think. Cool. Thank you very much, Garrett, for a fabulous presentation. I think I've used PyMOL for a couple of years, and I didn't know half of these things. So it's been really, really, very, very useful. Oh, good. I'm glad. Yeah. It's a really great program, and it's been great from the start. And I mean, Warren Delano deserves all the credit for believing in open source, making it available for free, standing by it, until he sadly passed. Yeah. Well, I think we've finished all the questions. We're getting a lot of compliments in. As I say, I think this has been a great first day, and we look forward to the rest of the week. So I shall be closing down the webinar now, and thank you to everybody who took part. Bye for now. See you tomorrow, hopefully. Thanks, Chris. Right then, bye.